All this is Dr. Mobeen Sayed, and possibly my assistant Luffy will be here soon as well. So, the discussion today is going to be about the Bacillus anthracis and anthrax. It will be in two parts. Today, we will talk about the introduction to the Bacillus anthracis, and then we'll talk about the properties and the pathogenesis of anthrax. There is an important part, and that is during the pathogenesis, we talk about the edema and gelatinous necrosis of the tissue that occurs, but exactly how the edema occurs is usually a very simplified statement, even for the physician documentations or literature. So, I wanted to make sure that we can actually see in depth how does the edema occur and how does the necrosis occur. I also want to make sure that we understand that the pulmonary anthrax itself does not damage the alveoli, but the damage occurs in a different mechanism. So with those possibilities, to know them, let's start a discussion. So this is drbeen.com. This is a very, very good set of uh, topics about anthrax. Unfortunately, I did not put the link in the description. What I'm going to do is I'll put the link right here in the chat. Then this is another very good reference from New England Journal of Medicine. Uh, then this is another very good study about the gastrointestinal anthrax. This is Wikipedia. Again, if you do not like Wikipedia, you can pick up this topic and read it from your favorite place. Up to date is great. Books are great. This is anthrax toxin. We'll talk about these things and 2001 anthrax attack. So with this, let's start a discussion. So today we'll do the part one, and tomorrow we'll do the anthrax disease itself and the management of the anthrax. So this is the Bacillus anthracis illustration. It is a gram-positive rod-shaped bacillus, and normally under a microscope, it appears like box cars, like a long train. It appears like small boxes or rectangles that are attached to each other. So let's say this is one rectangle, this is another rectangle, this is another, and so on. And this is why, because these pathogens, these bacteria are attached to each other in a line. Individual bacteria cannot actually move away. Neither does it have any structures on it, for example, flagellum, to have it move. It does have structures, but they do not help it move a lot. So we say Bacillus anthracis is actually a non-motile gram-positive rod-shaped pathogen. And down here, I've made some spores as well. One very important quality of this pathogen is it makes spores. It is the spores of this pathogen that can live for decades in harsh environment and can be used and were used in the US as a bioweapon. So I will explain that why is it wearing these glasses and why are there this little thing in here? Why is it flexing its muscles? What are these two red things? I will explain them in a few minutes, but this picture hopefully can stay in your mind. So Bacillus anthracis causes anthrax. Anthrax is a zoonotic disease. What does that mean? That it is basically a disease of animals. However, it can transmit from animals to humans. COVID, for example, is another disease where we have a pathogen that was supposed to be in bats and it has jumped to humans. Whatever was the intermediate or wherever it came from, that is not the discussion at this time. The important thing to understand is zoonotic diseases are those where the disease is originally of an animal species and then it jumps to humans as well. Now, mostly it is the Bacillus anthracis is found in the sheep wool or goat or horses and other animals. And as I said before, the spores of this thing have been used as bioweapon in the past. And we just discussed what is zoonosis. Zoonosis is where a pathogen that is on a animal jumps to humans and is transmitted to them. Interestingly, Bacillus anthracis has not been observed to transmit from human to human, but animal to human transmission is possible. Now, properties. This is important. It is a non-motile gram positive. So this color, this purplish color is a gram positive color. It is a non-motile gram positive rod-shaped pathogen. It can be aerobic or facultative anaerobic. What does that mean? Aerobic mean a pathogen that needs oxygen to grow. And then facultative anaerobic means that it can be okay without oxygen as well. Anaerobic means without oxygen. Facultative is it can be. So if there is oxygen, it will be fine. If there is no oxygen, it can still grow as well. There are some pathogens that strictly want to grow in the non-oxygenated environment, and then there are some pathogens that would like to grow in the oxygenated environment. So this guy can straddle both sides. It is spore forming as well. Spore forming, I would continue to say this because spores are the most important thing when it becomes really deadly. And it is a soil bacterium. So what happens is animal feces contains this pathogen, and when the feces are deposited on the ground, on the soil, then the pathogen and the spores can live there for a very long time. And from there, other animals, it can go to they can eat up, let's say they're eating grass, and these spores can enter their mouth and their GIT. Or let's say if a human being has touched grass or worked in the field and their hands are dirty and then they touch their mouth, the spores can enter their mouth. Or the dust could contain spores as well. So let's say if a human being is passing through some environment where there are spores in the dust, in the soil, and the soil is kicked up, that can cause the pathogen to enter their um, nose and mouth. This pathogen can actually infect the skin abrasion. So if let's say somebody has a skin wound on which the pathogen then settles, maybe a farmer who has a skin wound and is working in the field and the field has contamination, they can get cutaneous anthrax. Similarly, wool sorters or people who work with the animal hides and wools, they can either get inhalational anthrax, or if their skins have cuts and abrasions, they can get anthrax through, or Bacillus anthracis to germinate through those routes. So there are three types of anthrax. There is gastrointestinal, where a person has eaten something that is contaminated, or pulmonary or inhalational, where a person has inhaled the spores which are present in the air, or cutaneous, where the person has gotten some wound on which some debris is deposited, something that is not correct or trashy is deposited, and that may have the spores in it. Continuing, size. This is a very important thing. The size is, so it is a rod shaped thing, 3 to 5 micrometer tall or long, and 1 to 1.2 micrometer wide. Why this is important? This is small enough size that it can allow the Bacillus anthracis to go all the way to alveoli. Most of the time in our pulmonary system, those particles or pathogens that are larger than 5 micrometer, they are trapped somewhere higher up in the airways, and they do not get a chance to go all the way down to the alveoli, which is the end part of our lungs. But Bacillus anthracis is so small that it can go all the way and be deposited in the alveoli. And now here is an important thing for my physician friends. The Even when this pathogen deposits in the alveoli, it actually does not cause infection and damage there. It waits for the macrophages to eat it up. Macrophages act as a vehicle for this pathogen to spread it. So macrophage would eat it up, take it to the local lymph nodes in the pulmonary system, and there in the lymph nodes, it would start infecting these cells and start moving from there. So please remember this thing. This is a very, very important concept. Okay, so continuing. Genetic material. So the Bacillus anthracis, it's a bacterium, correct? So it has a circular DNA. So it has a DNA in the shape of a circle. That is its own DNA. In addition to that, these two little red things are the plasmids. Of course, we all know that plasmids are borrowed genetic material. These are borrowed powers. Like in the video games, you can get a power from somewhere. Here, the bacteria can actually exchange genetic material. They can give it to each other, and that is going to give them powers. Just like nowadays, let's say vaccines. Vaccines have genetic material that can be given to us, and then our cells have that power of making that specific antigen. Similarly, here, the bacteria can give each other as a gift plasmids or small pieces of the DNA. Those small pieces of DNA can have genetic material on them that gives bacteria some extra power. So in case of Bacillus anthracis, the virulence factors are the things that would help the bacteria cause infection and cause damage and cause disease. The virulence factors are present on these plasmids. So these two plasmids are called PXO2 and PXO1. On the PXO2 plasmid, it's a DNA structure, right? There is a gene for the capsule of the Bacillus anthracis. Now, Bacillus anthracis is unique. Its capsule is made up of protein. Normally, bacteria do not have a proteinaceous capsule. This bacteria has a proteinaceous capsule, or the capsule is made up of protein. That is why I made the pathogen to have these muscles. Why? I just wanted to depict that its outside is covered with proteins. Now that becomes a very difficult problem for our immune system. Why? Because when the poly D gamma glutamic acid, that is a protein name, when the poly D gamma glutamic acid is covering this pathogen and our macrophages come and try to eat it up, they cannot easily eat it. When they bite it, when they try to phagocytose it, it slips away because of its capsule. And when it slips away, it evades them, it escapes them, and that is virulence of this pathogen. That is one way it is poisonous to us, that we cannot actually catch and kill it. Then the second plasmid, PXO1. So Dr. Yo is here. Dr. Yo is saying, is it true Dr. Bean is running for president? That will be interesting. So yes, Dr. Yo, I will do that. So plasmid PXO1. So that the ticket will be Dr. Mubin and Dr. Yo. You can decide who becomes the president and who's the VP. Okay, so plasmid PXO1. PXO1 has two genes on it which are really important. Number one, lethal factor, and I'll explain these little later. So this is a gene that makes a protein that is called lethal factor. And then there is another gene on this plasmid, PXO1, that makes another protein which is called edema factor. There's also protective agent here as well. So there's a third gene over here which I missed to write, edema, a protective agent. Protective agent. Let me just write PA for protective agent. So how many virulence factors now we have? PXO2 has the proteinaceous material that is made for the capsule, and PXO1 has the lethal factor, edema factor, and protective agent. We'll talk about the protective agent in a second as well. Now, transmission and the life cycle. So here, so this is a sheep. So this sheep is dead. So imagine that an animal dies, and let's say animal was infected with the Bacillus anthracis. It dies, and of course, when the animal is dead, it is going to start rotting, and the bacteria and pathogens and other animals are going to start eating the flesh away. So slowly, the nutritional environment of the animal is going to degrade and go away. Now, if the pathogen, the Bacillus anthracis, was present in it, the Bacillus anthracis spores will come out. And those spores, down here if you see, these spores can live in harsh environment for decades. Decades. They can survive there. They can survive ultraviolet light. They can survive dry environment. They can survive heat. And they can sit there and wait for decades. Okay? So now when some other animal comes across these spores and accidentally becomes contaminated, and as I mentioned before, there are various route of entry that the pathogen, the spores enter that animal, or let's say there's a human being that again got the spores in the same way as I discussed before. The contamination routes are inhalational, GIT, and cutaneous. Then the spores would start germinating and the spores would start growing very, very sharp. So before we talk about how the spores would grow and the pathogenesis, very quickly how spores are formed. So remember we talked about it that these bacilli are actually made, they are formed in a structure of long chain. So this is one bacillus that would give rise to another, that would make another, that would make another. So those boxcar like structures will make a long chain, and that would become the Bacillus anthracis chain. If you may, can you see the ears of Luffy? So Luffy is sitting here. Okay. Now, how does a spore form? So look at this one over here. This on the left side, the pink one is, let's say, one Bacillus anthracis. This is another bacillus. Now what happens is one of the bacillus starts losing water from it, or starts becoming spored. Spore formation starts occurring. It starts losing water, it starts losing substance, it starts developing a hard shell around it, keratinaceous shell, just like a nails, not exactly nails, but hard shell like that. And then the mother bacillus, the bacillus near it, will engulf it and bring it in. So now one Bacillus anthracis is sort of pregnant with the other Bacillus anthracis that it just engulfed. It's an endosome now. And so that is the spore. So that means mostly Bacillus anthracis are running around. They're not motile, so they don't run around, but they're present with a tiny spore in them. They all carry a seed of them in them. This seed was actually another Bacillus anthracis that became a spore and went into them. That's very clever. And most of these sporulation occurs the same way. Now, spore germination. So what happens? When this spore finds its way, either through a skin wound or through inhalation or through eating contaminated food, and it goes into the GIT, these spores would start going to the local lymph nodes. How will they go to a lymph node? I'll explain that in a second. But when the temperature is 37 degrees centigrade, when carbon dioxide concentration is a little higher, when serum proteins as nutritional substances are present, and these can start helping the spores, you know, start picking up fluids from the environment, these spores are going to start germinating. They are going to start becoming the bacillus, and they would start opening up. So as soon as they open up, now they are deadly. And this mechanism was used by one of the doctors, or suspectedly used by one of the doctors in the US after 9/11. And he had sent out a number of letters that contained the spores. And those spores caused 22 infections, 11 cutaneous and 11 inhalational. I believe that he sent them through the letters of post office workers, and many other folks became sick. Out of 11 inhalational, 5 died, unfortunately. The FBI finally suspected the doctor, but before they could arrest him, he committed suicide. Now, incubation time, incubation period. Once the spores are attached to some body tissue, what time do they take to become germinated and start causing infection and symptoms? So, cutaneous anthrax, when the anthrax or Bacillus anthrax contaminates a wound, it can start germinating and start increasing and causing disease within 9 hours. However, it can be up to 2 weeks. So that means a person, let's say a wool sorter, a person who works with the animal hides or animal products, they may get contaminated and exposed 2 weeks ago and then develop anthrax a couple of weeks later. This is Davidson as a reference. Inhalational anthrax, 2 days. So if the anthrax or Bacillus anthrax goes through the nose or mouth and goes into the lungs, 2 days. And GIT anthrax can be 15 hours, can be 2 days, can be 5 days. So usually 2 to 5 days, but it can be as short as 15 hours as well. Now, my request to you, keep one thing in your mind, that most of the time anthrax starts becoming pathological, not on the tissue where it just contaminated, but mostly in the local lymph nodes that are near that tissue. So the question in our mind should be why? Why not right over there, but why the local lymph nodes? So that is what I'm going to answer in a second. It's a very interesting mechanism. So pathogenesis, and this is where we'll address this. First of all, we know that there is innate arm and there is acquired arm of the immune system. And the very first time when there is a pathogen that appears in our body, our innate arm would respond to it and start taking care of it. Dendritic cells and macrophages will eat up that pathogen. We know that from other microbiology discussions. And we also know that when the cell has phagocytosed the pathogen, that means eaten it up, that phagocytic vesicle inside the cell is then fused with lysosomal vesicle that has digestive enzymes in it. And when these two guys are fused, the digestive enzymes are spilled on the pathogen, and that reduces the pH, causes acidification, is sort of a stomach in the pathogen, in the cell, and that acidic environment kills the pathogen, digests it, breaks it down, and that is what we use. Not only we kill the pathogen this way, we take pieces of pathogen and then express them on our cell surfaces as well. Now, dendritic cell, when they eat Bacillus anthracis, they can very successfully do this function. However, when macrophages pick it up, remember macrophages have two problems. One, the pathogen has the proteinaceous material on it. And the second is when macrophage eat it up, they phagocytose it, and then they connect it with the lysosome, and then they reduce the acid environment or acidity, or reduce the pH, not acidity. They reduce the pH of the system, they acidify the environment, they try to digest this pathogen. Inside the macrophage, Bacillus anthracis actually becomes active in the acidic environment. This is why it is so deadly. One of the reasons. So when it becomes active in the macrophage, it will then come out of that environment, because instead of dying, imagine we take something, a bacteria, and put it in an acid to dissolve it, but instead of getting dissolved, it actually becomes active and now starts breaking things around and comes out of that little thing in which it was being digested. So it becomes active, it comes out of it. When it would come out of it, it would cause infection of the macrophage. Now here's the deal, this will not happen right away. Macrophage, for example, let's say, will pick up the pathogen from, let's say, a wound. Then the macrophage would run to the local lymph node to say, hey guys, look, I got something, right? That's a normal behavior of the macrophage. By that time it reaches there, a few days, a few hours later, while it was going there, it was trying to break down the pathogen, and pathogen got liberated. It infected the macrophage. Now it's going to break it down. So where would macrophage break down? The place where the infection occurred, or somewhere else? Somewhere else. Why? Because this little vehicle, the macrophage, was moving. And most commonly, where does it go? To the local lymph node. So where does infection occur? In the local lymph node. Why? Because by the time macrophages reached there, Bacillus anthracis has come out of the vesicle, infected the macrophage, killed it, come out of the macrophage, then infect other cells. This is why Bacillus anthracis, mostly the infections, start in the local lymph nodes instead of the place where it got contaminated, deposited. For example, in case of inhalational system, the lymph nodes near the nasal, oropharyngeal area and nasal area. If it is inside the lung, then the pulmonary hilar lymph nodes. In case of GIT, the Peyer's patches, the lymph nodes of the GIT are called Peyer's patches. The Peyer's patches is where the infection starts. If it is a skin wound, then local lymph nodes near that wound are the ones where the infection starts. This is actually macrophage's fault. It is the macrophage that is running around with this pathogen. This pathogen is immotile. It itself cannot run around. It's the macrophage that was going everywhere saying, hey, do you see this? I got something. And meanwhile, it would cause this whole issue. Okay, continuing. Now, this is important. Bacillus anthracis causes anthrax. Anthrax is usually a disease in which edema occurs. The swelling and the fluid extravasation occurs in the tissue. Interstitium is the spaces between the cells, correct? So the fluid increases there, plus the cells die, including macrophages that die. And that causes necrosis. Dead cells are necrosed. And a third problem is immune system is not working correctly. Why? Because you're killing the macrophages that are part of the innate arm. This is why this pathogen is such deadly. If it is outside of us, it can live in soil for decades. If it comes into our bodies, our immune system has a real challenge to work with this thing. So it has Bacillus anthracis has multiple virulence factors, as I discussed before. Now, first of all, let's look at the edema factor. So this is my depiction of an edema factor. From here, you should remember it. Edema factor is an adenylate cyclase enzyme. What does that mean? It takes ATP. So let's say this enzyme goes into a cell. Inside the cell, what is the energy system? ATP. It would take ATP and it would convert that into cyclic AMP. So one, the cell is now low on ATP. Number two, cyclic AMP has a number of functions that would cause the cell to become disturbed in its activities. So edema factor is a calcium calmodulin dependent adenylate cyclase enzyme. It's an enzyme. It goes into our cell, it makes more cyclic AMP. Now, what does that do? The cyclic AMP, you would read that in many, many books. Undergrad, grad, postgrad, and even in some studies. You would see simple message. Whenever cyclic AMP is increased in a cell, this would cause homeostatic imbalance that would cause edema. That's it. It would stop here. Whenever a cell has the Bacillus anthracis, that increases cyclic AMP inside the cell would cause the homeostasis, the water fluid homeostasis will become abnormal and edema would occur. This is usually what you would read. That is not sufficient for us to know. We're not average doctors, right? So how do we get that water out? So there are two processes that are very important to keep in mind. One of them you can find easily, the other one takes some time to research it, so I've done that for you. First is the cell itself can start losing water. And that is because of the cyclic AMP disturbing the cellular mechanisms, and I would explain them in a second. And so cells' own water retention and homeostasis becomes disturbed. Secondly, this is really important. This is a blood vessel I made here. In this blood vessel, imagine this is an endothelial cell, blood vessel wall cell. When this cell becomes infected with Bacillus anthracis, Bacillus anthracis edema factor causes the production of transendothelial macro apertures. What does that mean? Big holes in the cell. It helps create holes in the cell. When this cell, these are called TEMs, transendothelial macro apertures. TEMs are created. The cell makes holes in them. This is like if you take a plate and make a hole in the center of the plate. Now things from one side can go to the other side of the plate, correct? Now inside the blood vessel is what? Blood and fluids. You make a hole in the endothelial cell, which is making the wall. What would happen? Fluids would start getting out. This is the reason edema starts occurring. It's not just the inflammation causing edema. It is the Bacillus anthracis directly causing an issue to the endothelial cell, creating apertures in there, which then allow the water and fluids to move out of blood vessel into the tissue. This was really difficult to find, but found it. So that would cause edema in the local area wherever this infection is happening and wherever these blood vessels are infected. Now, why is it bad? Because once the patient starts losing fluids from inside the vascular system into the interstitium, the patient's blood volume and blood pressure is going to start going down, which would then lead to shock and death. So shock and death is not going to occur just because cyclic AMP caused its cells' homeostatic environments to be upset. The shock and death can occur because the endothelium starts becoming open to movement of fluids from blood vessel to outside. Okay, let's continue. So that is edema factor. Please don't forget this one. Next. Here is a macrophage. One more example with the edema factor. In the macrophage now, when the cyclic AMP increases because of the edema factor, the cell signaling of the macrophage reduces. This happens actually in all cells. Wherever the cyclic AMP would increase and ATP will reduce, that cell's signaling mechanisms will become impacted. Why are we then talking about macrophage? Because Bacillus anthracis sits in the macrophage. Why? Because macrophage, out of its innocence, ate it up to kill it, and the Bacillus anthracis came out in the macrophage and infected it. Now the cell signaling mechanism of macrophage is disrupted. When that is disrupted, macrophage's function will be disrupted. And when the macrophage is not working correctly, the pathogen would just roam around. This is like when the cats are away, the mouse will play. So the macrophage is away. It is sick macrophage. It's not working correctly. Innate arm is impacted. And not only this macrophage is now infected, but other macrophages are infected. So the immune escape is occurring. This is how this pathogen, how clever this is. This is why it is so deadly. This is why it is so difficult to manage it. Now the next enzyme, lethal factor. Lethal factor, what it does is, once it is inside the cell, it is a zinc-dependent endoprotease. Endoprotease, I hope from the word protease, you can see it is a protein-breaking enzyme. And it is zinc-dependent. It needs zinc to work with it. Once lethal factor, remember it is a lethal factor. Who is it lethal for? Not just for humans, it is lethal for cells. And when too many cells would die, then the person would die as well. So lethal for cells and then for humans, or the animal, whichever animal it has, livestock has that too. So zinc-dependent endoprotease. And the most important thing, in our cells, there is a protein called MAPK. MAPK is a secondary signaling system in our cells. Our cells are filled with signaling mechanisms. There are things that come and knock on the door with the receptor, and our cell internally does something. Or whenever cell is doing some function, there are signaling mechanisms that are working. MAPK is a very important signaling mechanism inside the cells. And what this lethal factor does, see, it is holding this little cleaver. It takes this protein, MAPK, and remember, lethal factor is a protease. It breaks proteins. It takes this protein and breaks its N-terminal. Remember, all proteins have a C-terminal or carboxy terminal and a nitrogen terminal or N-terminal. It goes and cuts the N-terminal. Of course, if you cut a protein, it's not going to function anymore. And here it is breaking down a protein which is important for signaling mechanisms. Signaling mechanisms are important for the cell to function. You think the cell would function now? No, this cell is going to die. So lethal factor snips MAPK. That causes MAPK related kinases to not function correctly, and the whole cell dies. Why does the cell die? Our cells have this mechanism in them that when they think I cannot function correctly, then they activate alternative signaling pathways, and they tell themselves to kill themselves. They cause apoptosis, they cause suicide. So when this pathogen disrupts the MAPK signaling pathway, the cell's sensors inside it, they look inside and say, oh man, I'm not working correctly. I am going to become a problem soon. I'm going to kill myself. And so the cell causes apoptosis and dies. That is the necrosis that is happening. That is the black eschar that would appear around the wound. We'll talk more about the anthrax itself tomorrow. Today, I wanted to make sure we talk about the pathogenesis. And finally, is the protective agent. Protective agent. This pathogen has 3 virulence enzymes, and that was edema factor, lethal factor, and the third one is a protective agent. And why did I make him look like this? Because without this agent, other 2 cannot work. And now, how do they not work? Here is what happens. Imagine this is a cell. We need lethal factor and edema factor, not we, the pathogen needs. We don't want it. Edema factor and lethal factor has to get into the cell to cause a damage. But how do they get into the cell? The cells are not just sitting open saying, you come in. So to bring these 2 enzymes in the cell, we need another enzyme that is given by this bacteria. This is called protective agent. What it does, the protective agent is following. This is, imagine this skull is a protective agent. It can bind to 2 receptors on our cells. One receptor is called tumor endothelial marker 8, TEM8. Other one is called capillary morphogenesis protein 2. These are just proteins or receptors, whatever their names, or CMG2. Protective agent can connect with them, bind with them. When it binds with them, like SARS-CoV-2 has TMPRSS2 that cleaves the spike proteins. Here, when this happens, furin proteins would cleave the protective agent and activate it. When the protective agent is cleaved and activated, it makes a number of protective agent proteins will come together to make a hole, to make a pipe-like structure in the surface of the cell. It is called a pre-pore formation. How clever that these protective agents, we are helping it. They come and bind with our receptor. We, in all of our innocence, we go and we say, okay, we're going to cut you and trim you and make you look nice. And then that little protein, 5 or 8 of them or so on, 7, 8, I do not know octamers. So heptamers and optamers, 6 and 8. I believe octa is 8. If not, so be it. They collect together, make a pipe-like structure, and make a hole in the surface of the membrane. But not just a hole. What they do, this is not a hole from which the fluids can move in and out. But now the lethal factor and edema factor can bind to this structure. It becomes a channel, and then through this, the lethal factor and edema factor go into the cell. All 3 of them will go into the cell. This is why it is called protective agent. If protective agent was not protecting the other 2 guys, the other 2 guys are broken down and not taken into the cell. It's a protective agent that creates this whole mechanism to bring them in. Once the proteins, lethal factor and edema factor, have translocated into the cell with the help of protective agent, then they would separate from protective agent and they would start doing their mechanisms. Normally, they stay stuck to the protective agent and become active as well. I think that is it for today. Right. So this is the discussion for today. This is a pathogenesis, very interesting pathogenesis, and there were many holes in many of the literatures that they did not fully put it. So I think this topic here that we covers a lot of important details. Tomorrow we will continue with this one, and we'll talk about the disease anthrax, the cutaneous, inhalational, and GIT. And then we'll talk about the signs and symptoms. We'll talk about the disease progression, the prognosis, and then we'll talk about the management of the disease. With this, thank you very much. And finally, if you would like to support this work, the smallest thing you can do, free thing you can do, is just like this video. Like, subscribe, and share. And if you would like to support this work, then there are links in the description. You can buy me a coffee or be a patron or use PayPal. Thank you very much, and I would see you tomorrow. We will not do chit chat. I'm trying to do the alternative day chit chats. So tomorrow we'll do the second part of this. Plus, we'll take up the chit chat as well, and we'll answer all the Twitter questions and we'll answer the live questions. So thank you very much. Stay happy, and I would see you tomorrow.